The ACAPELLA trial is a prospective observational study evaluating the frequency of serious infections and malignancy in a real-world population receiving Ocrevus, including those that were excluded from the pivotal Phase 2 and 3 clinical trials. We hypothesized that older and/or more disabled patients might have higher rates of adverse events. Since the FDA approval of Ocrevus in March 2017, data has been collected on consenting subjects that includes medical history, EDSS assessments, and data points of immunoassay as part of standard of care at our local laboratory. Adverse events were assessed real-time or during a patient's next scheduled infusion via questionnaires. Our total population consists of 306 patients since March 2017, where 62% of the total population has reached 1 year of treatment with Ocrevus, and 35% of all patients enrolled have had at least 2 years of treatment. Notable in our population is the inclusion of those who had a previous history of immunosuppressive treatment, older, higher EDSS, and immunoglobulin G levels lower than the limit of normal prior to initial treatment of Ocrevus. Disease breakthrough, moderate infections requiring antibiotics, and serious infections were evaluated across EDSS and age stratifications in our total population as compared to the parameters set by the Phase 2 and 3 clinical trials. 10 patients from the total population had disease breakthrough defined as clinical relapse and/or new MRI activity. The rate is similar to that observed in the clinical trials. An EDSS greater than or equal to 6 was associated with a higher risk of UTI. There was no increased incidence of infections, HSV, or zoster in patients with an EDSS greater than or equal to 6 and/or an age greater than or equal to 55. In our total population, 3% experienced a serious adverse event that required hospitalization. 3 patients developed malignancies of different types where malignancy occurrence is comparable to the rate seen in the general MS population. In our patients that have a pre-existing history of neoplasm, none have had a recurrence. 2 subjects became pregnant after treatment and both were unsuccessful events. The ACAPELLA trial has been interested in evaluating whether patients with higher levels of disability and/or age are at a high risk of infections, hypogammaglobulinemia, or malignancy with long-term use of Ocrevus. While we did see an increased rate of UTIs in our patients with higher disability, this is an expected finding within this population. There have been no other correlation of disability level or age with increased infections. Thus far in our cohort of 306 patients treated with Ocrevus for up to 3 years, our hypothesis of increased infections and adverse events in the older and/or more disabled populations has not yet been seen.